Bacteria are everywhere around us. On average, there are over 39 trillion bacterial cells in the human body at any given time. Although bacteria are often thought of as harmful, we would not be able to survive without them. You may remember from class that there are more bacterial cells in your body than human cells. Bacteria in humans actually help protect us from diseases, and those that live in our digestive tract help break down our food for more efficient nutrient uptake. The microorganisms contained within our bodies are collectively referred to as our microbiome. We learned in class that our microbiome is very diverse and provides a vital function in the breakdown and absorption of key nutrients, mostly in the large intestine during digestion. The relationship between animals and bacteria is so important that animals have evolved special organs for housing the bacteria that are needed for digestion, such as the cecum and the rumen in cows. Bacteria begin to colonize our bodies as soon as we are born. Furthermore, they are constantly changing as we develop and ultimately provide us with the framework for a mature, healthy microbiome. Many factors influence the array of bacteria present in our microbiome. Probiotics like yogurt help diversify the bacterial population, providing many benefits, especially absorption during digestion. On the other hand, antibiotics can negatively affect our microbiome by destroying both harmful and helpful bacteria. Lastly, prebiotics are a form of carbohydrate found in foods such as onions and garlic that cannot be digested, but rather serve as the food for the good bacteria contained within our bodies, promoting their growth. It's surprising, but the makeup of our microbiome can affect other things in our bodies besides digestion. This concept was tested in an experiment that studied the quality of sleep and response to stress as it relates to the bacteria contained in our microbiome. Stress produces a physiological response often referred to as fight or flight. That can result from a variety of different circumstances. As discussed in class, this is a response produced by the sympathetic nervous system. Stress and sleep are interconnected in that when stress levels are high, quality of sleep tends to decrease and vice versa. Sleep is a cycle with 5 different stages. The first 4 stages are collectively referred to as NREM, or non-rapid eye movement sleep, and the 5th stage is REM, or rapid eye movement, which is the deepest form of sleep when dreams occur.
Stress has also been found to produce imbalances in the makeup of our gut's microbiome, often by reducing alpha diversity, which is a measure of the number of different types of bacteria and how evenly they are distributed throughout a certain area. Certain probiotics, such as Lactobacillus rhamnosus, can aid in regulating brain function and the stress response by lowering anxiety and stress hormones. Previous studies have shown that negative effects of stress can actually be minimized by increasing the density of this bacteria. The researchers of this experiment sought to answer the question of whether consuming dietary prebiotics in the proteins lactoferrin and milk fat globule membrane found in milk can actually affect the body's response to stress and improve sleep quality by increasing the number of good bacteria in the gut, such as lactobacilli. They started their experiment with 24-day-old rats, feeding them either a diet full of prebiotics and the milk proteins or a normal control diet. Then they assessed the microbiome of the 2 different groups at several different points along their development, as well as their sleep patterns. The microbiomes were sequenced when the rats were 35, 70, and 91 days old, and the rats were equipped with EEG devices that measure their sleep and wake cycles at 59 days old. At 87 days old, the rats' tails were electrically shocked to serve as an acute stressor. Over time, lactobacilli was sampled from the rat species to observe its colony formations, and the rats' NREM and REM sleep were monitored to observe changes after the exposure to stress. To determine whether or not the type of diet had a statistically significant effect on the alpha diversity of bacteria in the rat's gut microbiome, 3 types of statistical measurements were used to analyze the data collected. Prior to the exposure to stress at day 87, there were no significant differences in these 3 measures between the rats of different diets. However, after the rats were exposed to the stressor, there is more diversification of bacteria present in the rat's gut microbiome that were given the prebiotic diet versus the control diet. This led to the conclusion that rats given the control diet had their microbiomes take a hit when exposed to stress. The researchers also analyzed data collected using the EEG device to find the correlation between stress exposure and REM sleep. There is no statistical difference between the sleep cycles in rats of different diets prior to the exposure of stress, but after the tail shocks, percent wake during the sleep cycle increased, diminishing both NREM and REM sleep in the rats of both diets. 20 hours after exposure to stress, the rats fed the diet rich in prebiotics had more REM sleep than those fed the control diet, indicating faster REM sleep rebound and longer NREM episodes. Overall, it was found that the rats fed the experimental diet high in prebiotics and milk proteins physiologically responded to stress better than the rats that were fed a normal diet. Those which were fed the control diet had reduced diversity in the bacteria contained within their gut microbiome and also had impaired sleep quality compared to the rats with a diet rich in prebiotics. In conclusion, the consumption of prebiotics reduced the impacts of stress in the rats. The results of this paper have very important implications, such as possibly feeding young children more prebiotics to reduce the negative effects of stress as they age. I bet now you wish you would have started taking prebiotics before you came to Georgia Tech.